Okay, there are 7 students remaining in this last group, and the first of those is Paige Oswald, and she is a third-year PhD student in cell and molecular biology, and she knew she wanted to study cell biology when she saw heart cells in a dish begin to beat on their own. That's out of that Frankenstein movie we heard about earlier. Okay. The title of her presentation is Heart Development, the Peanut Butter to Our Cardiac Jelly.
We all start out as a single cell. Then in the course of 9 months, millions of processes have to occur for us to develop into a complete organism. If one of these processes happens at the wrong time or at the wrong place, there are severe consequences in the form of birth defects. Heart defects are the most common birth defects in the world. And in the United States alone, 40,000 babies are born every year with a heart defect. So my research focuses on heart development and what might occur to cause these serious defects. The heart is such an amazing organ because it begins functioning before it's even done developing. So the movement of the cells contracting and the blood rushing through the heart influence how it grows. Surprisingly, you and a little fish called a zebrafish have a lot in common. Both types of hearts begin to develop in a very similar fashion. So I use the zebrafish to study human heart development. If you look at the embryonic heart, you will notice only 2 chambers, an atrium and a ventricle, separated by the embryonic valve. Within the embryonic valve lies a special cushion-like structure called the cardiac jelly. The cardiac jelly lies between 2 layers of cells, and it mediates that relationship between the cells. So for perspective, imagine you have a very uncomfortable pair of shoes. Your shoe and your foot have a very bad relationship. You're walking around all day, you hurt. But then I think of the cardiac jelly as one of those nice gel shoe inserts that you put in your shoe. So then you have a gap between your shoe and your foot, and they have a much better relationship, and you have a much better day. So we know this about the cardiac jelly, but there's still so much we don't know. So I focus on heart development and how the cardiac jelly may come into play. I have been able to change how much cardiac jelly is actually present within the heart. And by doing that, I've discovered a type of Goldilocks phenomenon. If there's too much or too little cardiac jelly, the heart struggles to pump enough blood to the embryo, making it very sick. So I've found and conclude that we have to have just the right amount of cardiac jelly to have a nice, happy, healthy heart. So with this research of this small yet really important structure, we can further understand how the heart develops. And with this knowledge, we have the power to detect and maybe even prevent these really dangerous defects, and we could save the lives of many. Thank you.